The overall goal of the following experiment is to measure the quantity of IgE-dependent mediator release from rat basophil leukemia cells. The allergy symptoms are caused by the release of mediators such as histamine. Now, histamine is not the only mediator being released by basophils and mast cells. The total of all mediators released by these cells results in the signs and symptoms of immediate and delayed hypersensitivity. Now, humans are not the only organisms that suffer from allergy. Dogs and horses suffer as well. In order to develop the research further and find treatments against allergy, such as vaccines, a release assay is required to quantify the amount of mediators released, if any, from the research treatment. This is the reason for this release assay. The RBL mediator release assay for the human system was first developed and published by Saraganian and Hook in 1986. It was later developed for the dog system and now for the horse system. The modifications are as simple as changing the proteins of interest. The cell line used was the rat basophil leukemia, or RBL2H3.1 cell line expressing the equine high-affinity FcεRI receptors alpha chain. To a cell density of 500,000 cells per milliliter, add the IgE antibody of interest to a final concentration of 1 nanogram per milliliter. Add 100 microliters of the mixture into a 96 soil plate and incubate for 16 hours at 37 degrees. This allows the cells to adhere to the well surface and the antibody binds to its receptor. Wash twice the media containing the remaining unbound antibody and dead cells with 100 microliters of warm 37 degrees release buffer. Discard the buffer from the last wash and replace with 100 microliters of the antigen of interest at the concentration.